Good morning, dear friends, ladies and gentlemen. It gives me immense pleasure to welcome you all to the 137th weekly webinar of AABN. AABN is now moving forward with optimism and an enhanced vision to create extraordinary value for entrepreneurs through enduring experiences for our guests, partners, and alumni, supported by our values of integrity, agility, efficiency, collaboration, sustainable development, and respect for entrepreneur community. On behalf of my colleagues on the core committee, I extend my sincere gratitude for the continued support of all the stakeholders. The resource person for today's webinar is Dr. Menaga Mahendran, Chairwoman and Founder, BioNeemtech India Private Limited. The topic for today's webinar shall be Look What's Fermenting, Science, Business and Future, an interactive networking webinar. Fermentation has been used in food production for millennia. Ancient civilization used microbial cultures to preserve food, create alcoholic beverages, and improve the nutritional value and bioavailability of foods ranging from kinch to tempeh. Over the past century, the role of fermentation has expanded far beyond its historical usage to a much broader range of applications. Fermentation now spans industrial chemistry, biomaterials, therapeutics, and medicine, fuels, and advanced food ingredients. The suite of tools developed through fermentation evolution is now poised to revolutionize the food sector by accelerating the rise of alternative proteins. As one of the oldest known sciences, fermentation technologies have played a large part in feeding the global population for thousands of years. More recently, fermentation has gained renewed interest from industry and consumers alike as one of the world's leading technologies to address issues regarding food sustainability and security. The link between fermented foods and overall health and wellness, and the potential to improve the quantity and quality of plant-based proteins and starches through fermentation are two of the major factors driving this renewed focus. In today's webinar, Dr. Menaga Mahendran will provide a global analysis of the rapidly accelerating fermentation industry. I have the privilege of welcoming and introducing Dr. Menaga Mahendran. Dr. Menaga Mahendran, the founder of BioNeemtech India Private Limited in women's of key start materials and intermediates for the pharmaceutical companies, chemical manufacturing sector using green chemistry methodology. She has trained more than 500 students for the past 9 years for the project work and academic skill development. She also gives guidance for the startups, women entrepreneurs for their conceptual-based ideas, product development, and support the product for commercial entry, startup to scale up. She has more than 15 years of experience in microbiology, enzyme technology, and drug discovery platform. Recently, she completed Goldman Sachs 10K Women Program and certified topper in Business Excellency in Indian Institute of Science, Bangalore. Added to the credits, BioNeemtech India Private Limited is a proud receiver of Sri Visvesvaraya Award for the Best Micro Industry in 2022 from Honorable Chief Minister of Tamil Nadu, conducted by the All India Manufacturers Association. She is an advisor to China Pharmaceutical Company, China, leading and supporting microbiology department for the screening assays of active pharma ingredients, intermediates as per international industrial guidelines, US FDA and TGA. She is an external expert of the Committee for the Adoption of National Innovation and Startup Policy at Sri Ayyappa College for Women, Nagarkoil. External industrial expert in Board of Study Syllabus for Microbiology at Manoramaniam Sundarana University, Tirunelveli, and an industry advisor member in the newly sanctioned Bio-IT Research Park, Center for Marine Science and Technology, MS University, Nagarkoil. Dr. Menaga's knowledge for the topic and her ability to engage the audience will ensure that attendees today walk away with several key takeaways. It will ensure that your attendance at the webinar today was more than worthwhile. I'm not going to make you wait longer. We'll let Dr. Menaga take over and regale you with her astute experiences and fresh ideas. Thank you, and over to you, Dr. Menaga.
Thank you. Thank you, sir. So thank you so much for this opportunity, Mr. Gopi sir and Mr. M.V. Subramaniam and other people. And I have to thank separately S.T. Gopi sir for making such a wonderful, pamphlets for the circulation. Anyway, so I'll go to the session because it was already time. Yeah. So today's topic, actually, I have made a very short slides of fermentation technology since it is a Business Alumni Association. I don't want to make much theoretical. I want to make a more interactive session. So yeah. So about the fermentation, these are all a small introduction. It's a process of growing microorganism in a nutrient media by maintaining some physical and chemical condition, thereby converting feed into a desired end product. So what is our desired end product? Based on that, we have to select the culture, the substrate, the medium, even the reactors. The fermentation technology is the use of organism to produce. It can be applied in the food technology, in the biopharmaceutical industry, as well as in the alcoholic beverages on a large scale of industrial basis. So that is the fermentation. So the basic principle involved in this fermentation technology is that the organisms here we are totally depending the fermentation technology we are totally depending on the bacteria or fungus or actinomycetes. We grow the organism under suitable condition by providing raw material meeting to also it has to meet the necessary requirements like carbon, nitrogen, salts, trace elements and vitamins. What is your desired product, whether it come out as a ethanol or whether it comes out as a antibiotics or whether it comes under some beverages. So based on the end product, you have to design the carbon you are using, the nitrogen, salt, trace elements, and some of the elicitors, and mainly the culture you are using. The end products formed as a result of their metabolism during their lifespan are released into the medium, which are extracted for use by human beings that have a high commercial value. So here, what is the major role we are playing is the microorganism is a factory. It's a mini factory. It is able to convert the feed into the end product, the desired end product, and it converts in a very mg level. But for our consumption but for our demand, it is needed in tons. So we have to design the reactor. We have to we have to make the engineering strain, and we have to design the media so that we will be getting the material in kilogram level. So that is the major thing of fermentation. So what are all the major fermentation products nowadays in globally? The demand product is, when coming to the industrial chemicals, the major product is ethanol, the lactic acid, and the organism produced is ethanol by Saccharomyces, that is the yeast, and the lactic acid is lactobacillus. This is the major two industrial chemicals. We are totally depending on many products. Enzymes, alpha amylase, proteases, and lipases. Overall, most of the industries like detergent industry or food industry or formulation, most of the biopharmaceutical, this depends on the alpha amylase, proteases, and lipases. So coming to the antibiotics, you all know about the penicillin, the overall, antibiotic, the first generation, from the first generation. This is said to be the first generation antibiotic, penicillin, and streptomycin, chloramphenicol, everything. Even now, we are not depending on the synthetic route. We are only depending totally on the fermentation-based product. We are depending on the cultures like Penicillium, Streptomyces, and species for producing these antibiotics. And vitamins. Coming to the vitamins, some of the vitamins like riboflavin and vitamin B12, this is totally depending, we are depending on the cultures for producing this end desired product. So these are all the major fermentation products. We depend on the cultures. We can't go with the synthetic route. So again, what is this introduction is, what is the difference between a bioreactor and a fermenter? So when coming to some point, people will be get confusing. What is a bioreactor and what is a fermenter? A bioreactor is defined as an apparatus such as a large fermentation chamber for growing organisms such as bacteria, yeast that are used in the biotechnology manufacturing of substances like pharmaceuticals, antibiotics, vaccines, or for the bioconversion of organic waste. That is the bioreactors. They have a separate parameters. The fermenter can be defined as an apparatus that maintains the optimal condition just for the growth of the microorganism used in large-scale fermentation and in the commercial production of antibiotics and hormones. There are some typical difference between the bioreactor and the fermenter. So what is common in bioreactor and fermenter? In both the fermenters and bioreactors, fermentation is carried out. That is the common one. And fermentation can be an anaerobic one, or the fermentation is an anaerobic conversion of sugar to carbon dioxide and alcohol by yeast. So this is a common parameter between the bioreactor and a fermenter. So coming to the fermentation or reaction, normally it falls in only two categories, whether it is an aerobic character or it's an anaerobic, whether it need oxygen, whether the culture need oxygen or it comes under, without oxygen, it has to be grown. So for aerobic reactor, it need adequate mixing and aeration for oxygen supply. So some of the end desired products will be in an anaerobic condition. If you are growing the culture, anaerobic culture, in the anaerobic situation, it will not produce your desired product. So no need of sparging. Sparging is the supply of oxygen in the fermenter. So no need of sparging or agitation in case of anaerobic bioreactor. So the right side, I have given an overall, design of a reactor, how it could be. It has a motor for the, it has an acid base for pH control. There are some parameters, it will come one by one. In the fermenter reaction, it has to be maintained. The pH should be a major, pH is the major parameter of a fermentation. Steam used for sterilization, everything, the fermentation production should be in a sterilized condition. The foam, if there will be always the formation of foam because we are using the protein substrate. When the protein substrate is used, there will be the foam and making production of more foam will be entering into the cutting of the desirable production. So that we need to use some anti-foam. So foam beaker, flat bedded impeller, culture broth, and last one is the sterile air. How to this is the overall design of a fermenter. This is a cross section of a fermenter. This is mainly used for the penicillin production. You can see that how much complicated. Normally for a production of an antibiotic, we have to design the reactor. So it also needs an antibiotic, acid base in, antibiotic out. Any reaction, fermentation reaction, when you start, it cannot be interrupted in between. You have to take a sample in between and you have to understand whether it is an acid condition or if your reaction should be in acid condition, you have to maintain the acid acid pH throughout the reaction. So you have to take the sampling out and check its pH and the culture stability, whatever you have to check it. So for sampling out should be there, and nutrients in, sterile air in, gases out, stirrer paddle, everything. This is the cross section of a reactor. So coming to the types of fermenter, when coming to the fermentation technology, I already said we are totally depending on this microorganism, and that all the reactions should be carried out in a particular aseptic condition. And what are all the types of fermentation? There are some simple fermentation, like normally in the lab, we carry out with the batch and the continuous reaction. Batch reaction, as already that name itself, you can understand it. It's a batch reaction. You have to start a reaction and stop a reaction. Continuous reaction, you can start a reaction. In between, you can feed some nutrients, and then it's a continuous reaction. Then fed batch fermenter, airlift fermenter, cyclone fermenter, tower fermenter, and there are also more other advanced systems. These are all some of the basic different kinds of fermenter I have just mentioned to understand. So here I have given you some of the different, what is the stirred tank reactor, airlift reactor. So this is for sometimes, you can't go for a common reactor for all the antibiotics. For each and every antibiotics, there needs a customized, very peculiar parameter applied thing. So coming to the stirred tank reactor, applications include the production of antibiotics and free immobilized enzymes. For the free and immobilized enzyme catalyzed reaction, we can use this stirred tank reactor. The airlift reactors, its mixing method is by the mechanism is by the airlift. So what are all the compounds you are giving? There is a homogenization of all the components present in this airlift reactor. It has a central draft tube, upflowing stream and downflowing stream. The homogenization of all the components is present in this. The application include the bacterial, animal, plant, fungal, and yeast cells. So this is the other one is the bubble column reactor. The mixing method is gas sparging. Sparging is in the bottom. You can see a sparger where we supply the oxygen through this sparger. So this is a simple design. It is used for a good heat and mass transfer rates. Low energy input and gas liquid mass transfer coefficients depend largely on bubble diameter and gas holdup. The next one is the packed bed reactor. Normally, most of the people used this for using the biocatalyst column with an attached biofilm. You can see a packed bed column in the side. So pump is required to make fluid move through the packed bed. Through the packed bed, we make the column. It's like a column and it contains a packed bed reactor. Application is mainly used in wastewater treatment. This is also another way of, the fermentation where using the wastewater treatment, it is used. Fluidized bed reactor. When the packed beds are operated in upflow mode, the bed expands at high liquid flow reacts due to upward motion of the particle. Energy is required. This is also used in the wastewater treatment. The fluidized bed reactor is all used in the wastewater. The tickle bed reactor, this is another type. You can see the liquid is sprayed onto the top of the packing and trickled down to the bed in a small rivulets. In this process, the gaseous pollutants on the surface of the carriers is absorbed and immediately biologically degraded by the microorganism. These are all used in the wastewater treatment. So coming to the different, you can understand what are the different types of fermenters used for our product. So what is the parameters mainly we have to focus on this fermentation is, because I will come at the last 2-3 slides, we will discuss about what is the major global demand of the fermentation based technology and why we are not up to the level, why India is not up to the level of the fermentation technology. The parameters mainly we should consider when you are entering into the fermentation technology is the power consumption. Power consumption should be as low as possible. Otherwise, you can't bear the cost. The commercial cost, you can come out with the commercial cost. So power consumption is the major criteria in the fermentation industry. Temperature control and pH should be provided. Sampling facility should be provided. These are all some of the parameters that has to be included in the fermenter. From evaporation, losses from the fermenter should not be excessive. The minimal use of labor in operation, harvesting, cleaning, and maintenance, because so much of laborious work will spoil the fermentation technology, because you all know that everything should be handled, everything should be done in a hygienic condition. So only technical experts can, enter into and do this. Should have an internal smooth surface. Containment involves prevention of escape of viable cells from a fermenter or downstream equipment. Aseptic operation involves protection against contamination. These are the major key points, parameters when we are operating a fermentation industry. So it's a classical biotechnology. Fermentation is said to be a classical domain of biotechnology. The humans have been using this technology for centuries. So it involves harnessing of the wastes of bacteria or yeast for the products of human we consume. It's a big business. So coming to the, when I say it's a big business, yes, the next one is, it is the major platform for producing the antibiotics. The compounds, antibiotics is nothing but the compounds that kill or inhibit the growth of other organisms. The most of the antibiotics are produced by filamentous fungus or actinomycetes, and they're derived from special organism or other living system. These are all very, over 10,000 antibiotic substances have been reported throughout this fermentation process. Any large-scale microbial process occurring with or without air is for fermentation. The process may take a few days to obtain an extractable amount of product. The antibiotic production is normally done by the batch process. So when coming to the business point of view, the antibiotic is a big domain. Now the whole world is operating for the production of these antibiotics using this fermentation technology. So other than that, the products also produced by the microorganism is enzymes, antibodies, steroids, alkaloids, food additives, ethanol, and citric acid. So coming to the microbial products, there are two types of metabolites. Normally, you all know that microbes produce in two types of metabolites. The first one is the primary metabolites, and the secondary one is the second one is the secondary metabolites. The primary metabolites also include mainly the vitamins. I have list out all the vitamins produced by the organisms, like thiamine, riboflavin, pyridoxine, folic acid, biotin, vitamin B12, ascorbic acid, beta-carotene, ergosterol. These are some of the primary metabolites, that is the vitamins produced by the bacteria. and we are using for our all type of products. It can be used in the food industry. It can be used in the healthcare industry. It is used in all the type of biopharmaceutical and pharmaceutical industries for making these vitamins. Without vitamins, there is no healthcare products. You all know that. So all those key ingredients that is, we can say, as key raw materials, that is only produced by this bacteria using this fermentation technology. So other than that, you can also see the industrial product is beverages, antibiotics, organic acid, amino acids, enzymes, vitamins, organic solvents, SCP, single cell protein, steroids, vaccine, pharmaceutical drugs, and some of the dairy products is also produced by this fermentation method using the microbes. So coming to the commercial production, why it is not a simple thing? Yes, it is a complicated process. We can't just like biological method of production is a very complicated process, because some of the commercial production, like say for example, I have given here cyanocobalamin. The production of cyanocobalamin, it is not a single step process. It involves different nutrients, medium optimization, medium design, as well as the culture, engineering culture, and also it involves anaerobic as well as aerobic cultures. Two different types of fermenter is involved here for the commercial production of cyanocobalamin. So that it's a complicated process. The other one I have given is vitamin B2, riboflavin. It is an water soluble. So it is a very simple process, normal simple process. So ascorbic acid, I have listed out some of the very important like cyanocobalamin, vitamin, riboflavin, ascorbic acid, because most of the industries, largely the biopharmaceutical and pharmaceutical industry, is depending on making their product this important type of vitamins. So ascorbic acid is mainly used in the collagen biosynthesis. It is protects against, these are all some of the common thing I have given here. You can see the right side, you can see the process, how to make the L-ascorbic acid of 100 grams. You can see we are using so many, it's a co-culture type. The other thing is there is two types of fermentation method. Whether you depend on a single culture to produce a particular compound, or whether you depend on some co-cultures. One or more than 2-3 types of cultures can be joined together to form a desired product. So this vitamin C comes under a co-culture method with a submerged bioreactor fermentation type. You can see the species involved is Irvinia, Acetobacter, Glucanobacter. So you can see that 2-3 more than one culture is involved in the production of this L-ascorbic acid. It is also a very complicated fermentation-based product, now have a big market demand. Apart from this, vitamin C, the another primary metabolite is the organic acids. The organic acid is also like butyric acid, formic acid, propionic acid. These are all malic acid. These are all very important organic acid. Major market demand organic acid in not only in India, overall globally, it is a very demandable product. We are totally depending only on fermentation technology for producing this organic acids. Citric acid, it may be a very simple thing we are saying, but it is applied in flavoring agent, in chemical industry, in pharmaceutical industry, in the detergent industry. It is used as an acidifier. It's used as a flavoring agent, chelating agent. It's a primary metabolite present in all the, you can take it from bacteria, some of the organism for making the citric acid. Citric acid and acetic acid are plays a major role, a market demand product nowadays. Can see the different types of acetic acid used in as the industrial uses. Different forms of acetic acid we are used in different types of industrial thing. So coming to the secondary metabolite, those what I explained is the primary metabolites. The secondary metabolite is nothing but the antibiotics. So the antibiotics is a broad spectrum and narrow spectrum antibiotics we can produce using this fermentation technology. The major market demand is streptomyces, that is the tetracycline and actomycin D. These are all some of the very important antibiotics. Now we are having a major market share, market demand. So some of the antibiotics produced by the fungus are beta-lactams, penicillin, cephalosporin. So produced by prokaryotes also, I have given here aminoglycosides, macrolides, that is the lactone ring bonded to sugar. This macrolides is a very, big form of huge market demand product nowadays. Tetracycline, peptide antibiotics, as well as platensimycin. So, I have explained you so many types of antibiotics, so many organic acid, enzymes, vitamins, everything is produced by the bacteria, fungus, actinomycetes. We are totally depending on the fermentation technology. And what is the business? What is the future? Or what is the business of this fermentation technologies? When you come, we can see, I have listed out here, what is the market US dollar in billions. So coming to the cephalosporins, it occupies about 9.9 US dollar billions of market share. Penicillin 8.2, other beta-lactam is 1.5. Antiviral including vaccine is 10.2, quinolones, antifungals, aminoglycosides, tetracycline, other antibacterials and other anti-infectives. These are all totally market occupied in $billion. And this everything comes under, we are depending on the microorganism. Using the fermentation technology, we have to make this. You can see the global microbial product market also. In 2022, it is around US dollar is around $242 million. In 2022, it is forecasted that in 2027, it will be around $346.3 billion US dollars. This is only based on the fermentation technology. So here also you can see the global fermentation chemical market size was evaluated as 79.84 billion in 2022, and it expects to be around 134 US billion dollar by 2030. It was also a research. What is the fermentation chemical market? This is only based on the fermentation chemical markets. So the same, it was given in different, where are all, the leading people are there for the fermentation chemical markets by region. So North America, Latin America, Middle East, Africa, Europe, and Asia Pacific. When you are taking Asia Pacific, it is showing the China. So there are so many, major key players are in these areas for, they are holding the major fermentation chemical market share. This is a report given in 2021. So overall, the projected period, it is anticipated that rising per capita middle class disposable income expansion in the food and beverage sector in nations like China and India will boost regional demand. So coming to the fermentation products, who are all the key market players globally? Novozymes, Ajinomoto, BASF, ADM, Cargill, AB Enzymes, DuPont, DSM, Lancetec, Microbioform, Novacep, Biocon, Avinoc, Dow, Lonza, and these are all some of the key market players of fermentation-based products globally. So different segments are also here. This is a very summary I have given here. So coming to the segment, the fermentation-based products, it falls under three segments. A product, coming to the products, it be the end desired product can be applied in the alcoholic industry, the enzyme industry. The alcoholic industry is the major share. It holds around 52.5% shares. In alcoholic beverage industry, we are using the strains for producing alcohols. The enzymes is around 15%, organic acid is around 25%, and other products is around 6.5%. So coming to the segments, normally the application part, it is used in the industrial application, food and beverages, nutritional and pharmaceutical, plastics and fibers, cosmetic and toiletry, and other application. And coming to the region, North America, Europe, Asia Pacific, Latin America, and Middle East Africa, these are all the major, you can see the players of this fermentation-based product. So a recent market development is, these key players, they are more concentrating on their R&D. They have a very strong R&D system, and they develop, say, for example, Novozyme introduces Innova Apex and Innova Turbo, two cutting-edge yeast that reduce the need of for pricey urea and boost ethanol yield without compromising resistance to common production stresses. So everywhere, the major, you all know that as a business, people, you all know that we have to think about the cost cutting, the commercial production of some particular molecules. So whether we have to concentrate on the media design, or we have to concentrate on some, engineering strains or something else. So these are all recent market developments. Novozymes, then two novel bio-based surfactants from Zolway, and the collaboration agreement was between Novozymes and Novo Nordisk Pharmatech, combines the business shared interest and expertise in creating specialty enzymes. Because the other one is, everyone is now specialty enzyme. What is a specialty enzyme? It is mainly used for the biotransformation. Say, for example, most of the chemical companies, you all know that people are using in a very conventional method of producing some molecules, but it needs a large amount of hazardous chemicals, solvents, everything, catalyst, if it is a chemical catalyst. But coming to the point, if that particular catalyst is altered or replaced by the biocatalyst, definitely you can have all the catalyst enzymes are species specific or reaction specific. So the yield is more, as well as we will be able to, eliminate all those hazardous chemicals, solvents, man safety is there. When we are replacing all those chemicals using these enzymes as a catalyst, definitely there is a big revolution in the pharmaceutical industry. And now people, big people like Novozymes, Merck, Pfizer, they are more concentrating on replacing the catalyst using the biocatalyst for making the reaction specific, as well as the wastage is also reduced much more there. So everything, these are all we can only concentrated using fermentation technology. So this is a very important slide I want to show you. I took this from the Government of India website. You can see that these are all some of the key, nearly 22 APIs. That is the key critical APIs. They say as key critical APIs. We are depending mainly on the imports. We are not using these molecules. The local players are very low, very less in India. Around, you can also see that the percentage of import we are depending. Azithromycin, the percentage of import, sorry, yeah. Meropenem, vitamin B12, rifampicin, acyclovir. These are all some of the around 22, around neomycin, clindamycin, streptomycin, gentamicin. These are all tetracycline, vitamin B1, everything, claromycin, everything is fermentation-based products. It was actually displayed in Government of India website. They have clearly given that these critical APIs, that is fermentation based products, we are totally depending on other countries. It is totally depending on the imports. We don't have our own particular technology to make this in a commercial production, not in the lab research. Lab research papers, patents we are very good in lab research papers and patents, but commercial production, we are depending totally on other countries for this, particular 22 key start material. And the Government of India is also supporting us. If you are able to make a technology and produce, start a manufacturing unit for making this product, they are giving around, more than 30% of capital subsidy for making or producing this. And they have also production based some schemes also there for supporting us. So this is a major, when coming to the fermentation based product, not only food and other beverages, the antibiotics, this is a very critical area. This is a very big business also. We have to, as a microbiologist or chemical engineers or mechanical engineers, because this is like a team of experts joined together to come out with an engineering strain. With only making an engineering strain by a microbiologist or biotechnology cannot come out with a successful commercial production. It has to be, tie up with some chemical engineers to design the reactors. It has to be, tie up with some mechanical engineers to, support the reaction condition to be, the reactors has to be very customized and productive. So for coming out for the commercial production, it's a team of experts should be joined together. You can see how much critical APIs we are depending on outside country. It's a very dangerous zone. We can't always depend outside for the healthcare products. So that's why this is a paper article I have given here in 2021. It was by some of the experts says that the fermentation products manufacturing may need a little more push than the PLI scheme, because they have clearly said, he can see that at present India lacks in technology in this area and needs to outsource the technology part of these projects, and the cost of technology, uninterrupted power and water in abundance. Cost of power will have a major role in winning this business. Why I have given here is, even though we are talking more about the fermentation technology, where we are lacking, the lacking is we don't have a particular customized, technology as well as things. I just want to give here. The investment in fermentation based products is much higher when compared to the chemical synthesis. There are hardly a dozen top Indian companies who are capable enough to venture into manufacturing fermentation products. Out of the cost of fermentation based products, almost 40 to 50% is the cost of power. The environmental requirements are also another challenge. These factories can be set up only near the sea, considering the humidity in the coastal region is more. Fermentation based products cannot be kept in humid atmosphere. So like that, there are so many parameters they have given here. They want to push up the fermentation based products globally because it has a big demand. The global demand of the fermentation based products like antibiotics, enzymes, organic acid, especially the specialty enzymes, specialty chemicals using this fermentation based technology is very much. So we have to more concentrate on this. So this is my, short slides about this. I will also explain about my company. Then we will start an interactive session. My company, our company, Bionim India Private Limited, already he has given a good intro. It's an 9 years old company. It is in Women's Biotech Park, Sirseri. It's a kind of, lab facility. We have a continuous flow chemistry facility. We are totally working on, we are synthesizing molecules using the green chemistry methodology that is already endorsed as a zero carbon emission. We are also supported by MSME for making key start material using this green chemistry, as well as we are the Biorac Seed Fund winner for making the key start material using the green chemistry methodology. For making the molecules using this, safety green chemistry, we are also a proud receiver of Sir Vishweshwaraya Award from our Honorable Chief Minister of Tamil Nadu, Mr. M.K. Stalin in 2022. Is conducted by All India Manufacturing Association. This is my company history. Now we are having a commercial production for making we are entering also into the commercial production using a GMP facility. We have a tie-up and we started working on commercial production of our molecules. So our team expertise is, we have our R&D center, we have a pilot plant facility. So if any molecules are with you, we can go for the lab validation and we support for your innovation and patents. We also synthesis molecules using the pharmacopoeia as per the pharmacopoeia standard. We support for DMF as well as we support you up to the commercial production. So these are all some of our, various domain of Bionim Tech. So process chemistry, chemical engineering, fermentation, enzyme technology. So we support for people for making vitamin, amino acids. This I have given here. This is my pamphlet actually. The probiotic is a very important domain. We are working for making some nutraceutical and probiotics. So bioactive components. We have a handful of, we are tied up with some international clients for supporting them for the technology development for API and nutraceutical intermediates. So we are very strong in making molecules using the green chemistry methodology. We have come out with 2 important patents also for making the molecules using the continuous flow chemistry. So we have a pilot plant with all those facilities. These are all some of the, patents and key publications of us. Our director, Dr. Mahendran Balachari, who has around 30 years, more than 30 years of API industry experience for designing and developing the molecules using the chemical synthesis. So this is our portfolio of Bionim Tech. So this is our international collaboration for taking up, our molecules, both the chemical as well as the biological method, from gram scale to the kilogram level, and then up to the DMO filing. So very thing is, very soon we are entering into the commercial production of in the GMP plant also. Thank you.
Thank you, ma'am. Thank you for the nice session.
Thank you, sir.
Yeah, so we'll keep the floor open for question answers now.
Please.
Participants, you may please unmute and ask your questions, please.
Somebody has asked some
Madam,
question. Yeah.
Hello, this is
Yeah, please
Venkat
go on.
from
Yeah,
Tirumala Chemicals.
Hello, sir.
Ma'am, thank you for a very interesting presentation. As a person who is involved in the chemical industry, I have seen some commercial transformations happening through the bio route. And you mentioned rightly some examples: citric acid, acetic acid. We make malic acid and thermal chemicals, but we make by the petrochemical route. But recently, some Chinese have started making what is known as L-malic acid from fermentation routes, and they claim to be competitive with petrochemical route. But one of the challenges I've always found in fermentation routes for chemicals is the power cost, which you rightly mentioned, that power has to be very low, and the steam and the evaporation cost, because invariably these are conducted in a water medium, and you need to evaporate a lot of water from at low concentrations to high concentrations to isolate the product. So can you throw some light on latest developments that has changed this notion?
No, this is actually now, this I have presented because these are the key parameters where we have to think about and come out with some solution. Even now, I am doing my fermentation. We are working on some macrolids synthesis production using the fermentation. So here, these are all the parameters where we are also meeting the challenge. So we are expecting how we can solve out. By designing the reactors. I think, as you said, my suggestion is by designing some of the customized reactors, we can able to meet some challenges.
To give some numbers, madam, citric acid, the product is about 60 to 70 rupees a kg selling price. So just the best I have seen, looked at some fermentation technologies available globally by technology suppliers. They are talking of a power consumption of about 2,800 to 2,900 kilowatt hour per ton of these products. So if you use a power cost of 8 rupees, automatically the power cost itself is coming to about 50% of the product cost.
The
and
only
in
option,
China,
yeah,
it is much lower.
Yes,
Because China has a power cost of about 4.5 rupees, whereas we are at 8 or 9 rupees. So unless we bring down the entire citric consumption, about 80,000 tons in India per year, is being imported from China, mostly from China, 90% from China. So how do we as India, be Atmanirbhar in citric acid? I mean, given that our power costs cannot change much.
That's what, sir. The only alternative we can make the power cost reduces, like one time, like solar. We have to, think about utilizing the solar power plant for making all those factories. But still, it is an one-time investment. We have to get support from the government at least to install the solar power plant so that we can cost cutting. Yeah, as you rightly said, the 50% is 40 to 50% of cost is totally depending on the power. So we have to think as a green energy source, some green energy source like solar plant. Like China, why China is so they are able to meet the challenges and give so much low prices is they don't depend on their EB price is very low because most of them they are making on the solar. They are making so much of solar plant, and they are giving good enough to the factories. They are supporting, the government is supporting good enough to the factories. So like that we should also to be, our government also support us in making this. That's why I made that paper article here. Without government support, we can't give a product in a very, low price like China.
Yeah. So, I mean, just if I'm just leaving this debate open to all the participants here, that there is a very good business case of citric acid, provided we break this economic barrier that I talked about. There is additionally steam. Steam is about 8 tons or 9 tons per ton of citric acid. So, at a cost of about 3,000 rupees through furnace oil generation, this is what most companies do for generating steam. It's, I mean, with this economics, you can never compete with them. So this is the issue, and it's true for a lot of products like citric. But I'm sure somewhere, if somebody can break this barrier, terrific business case. I'm just leaving it open. Suggestion.
Yeah, Mr. Venkat, I'll take a lead from you. And thank you, Doctor, for your intense, informative session. And you are into innovative area. Only I just needed to understand what you mean by green technology. Okay, I know this thing, but I'll continue with now Mr. Venkat's thing. I was having certain analytical data. Mine would be maybe about 80-90% right. I have been one of the failed model of a penicillin industry in India in 94, 95. There are many penicillin industry have come up in India and all have dropped out their manufacturing process because of the competition. And so my data or my information based on the exposure which I have got with the penicillin. Of course, in the food industry, I had an exposure about the enzyme, okay, and which continue to be flourishing. But in case of a fermentation industry, I take it as 1/3 of the cost is power. And 1/3 of the cost is the interest and taxes. And 1/3 of the cost is only the raw material. Those time in 97, 98, 99, when we compare the RM cost of the Chinese manufacturer of a penicillin and us happen to be not more than 5-10% difference. Though they have improved the productivity, but we have well managed, all the Indian companies have well managed the nutrient management much efficiently, and then we could able to compete their raw material price. But interest and tax for 1,000 mega million units, the cost those time was about 250 crore. The 250 crore can be investment. So of which maybe 50% they have gone for public issues, somebody have gone for loans. So the cost of the interest have come down by occupying our sheet around 1/3. So this is where probably this recent one, okay, they are giving an incentive by the Indian government for having these industry require a lot of investment. So this definitely needed a long-term kind of a support to have this 1/3 interest and the taxes benefit from the government. If not, nobody else can help. Otherwise, maybe it becomes not a business, it becomes a charity. Now I'll come back to the third part of the power. It is an aerobic fermentation process. So if I really divide the cost into the three, 30% I said it is a power cost, of which more than 50% of the cost means around 15 to 18% of the cost is only for the bringing the temperature of the fermentation technology, because most of the fermentation is at 25 degree and we are in tropical countries, our temperature is 34, 35 degrees centigrade. And from 34 to 35 to bring down to 25, maintaining it, because our atmospheric air is also at 34, 35. So 20% of the overall cost is only on maintaining the chilling. If I refer back, okay, the Chinese manufacturing industries, they have set up most of their penicillin G plants in a chilled condition area. They don't have anything. This 20% of the cost is grossly coming down, except the extraction crystallization process, which they have to do. But otherwise, for the fermentation, their energy spent is almost nil. That is the thing where if we need to really improve the fermentation technology for us, my request to Dr. Menaga is probably we may have to develop strains or a technology which works at 30, 35 degrees centigrade to come over it. If it is going to be at 25 degrees centigrade, this 20% cost is going to taken away. Maybe it is through solar or it can be anything. Okay, but that is going to take place. That's what is the development needed is fermentation at a higher temperature. When I say higher temperature, maybe 30, 32. Yeasts are growing at 30, 32. We may have to, India, for a tropical country, for us, the research for the fermentation should be, even if you make it 28 degree, even if you make it 30 degree, but that time we were not having an R&D. So we have tried a few trials at a higher temperature, but our strains did not work for it. It gone into the back. So the challenge to the R&D or a development is improve or increase the temperature of fermentation could be one opportunity, and the government to support interest and taxes considerably for a long duration, not for 1-2 year. It has to be for 15 years because this is a high investment and low return product. Though it is the quantity is bulk, the percentage of return gross margin cannot be more than 10 or 15%. So these two are looking to be a potential opportunity or to look for in the research and the support system. Thank you.
Fantastic, sir. Nice input. Thank you so much, actually. Yeah.
Thank you. Yeah, very nice.
The question now is, yeah, you are right, sir. We have to develop the engineering strain because I will also share some of my experience. Now we are developing some strain for making the macro lids. It is purely for a I have knocked doors of DBT, DST, so many, I'm not blaming, I'm just saying, because getting grant support from the government is not an easy one here. Now we have got an international project and we are developing the strain because it is totally supported by that particular client we are working. So as you said, if I want to develop a strain that is able to produce the desired product in particular temperature, it needs a lot amount of R&D support. So if government is able to support R&D, it cannot like asking the question, did you finish the proof of concept? Did you go for a publication? What is your TRL level? So for reaching the TRL level itself, we need the R&D support, grant by funds and grants. So government should look over on this and they have to support. As you are rightly said, the power consumption is the major thing. I will also share one news here. I don't know whether you are aware about this. Only for making the fermentation based product, central government has sanctioned a big project in Dindivanam in Tamil Nadu. It is a central park facility, research facility for making the fermentation based product. Just they have sanctioned and started the work 3 months before, I think so. So I think this is a very good news, at least for us. They have started a central facility park for making the fermentation based product. So that I think if it is a central based park, we will get a good subsidies or something else. I don't know how it will come out. The park is sanctioned now. It is open to us manufacturers.
Okay. Thank you. Thank you. Thank you, Doctor. Thank you.
Yeah. Thank you, sir.
Preeti had a question. Preeti, you can please ask Preeti.
Good morning, ma'am.
Good
Today's
morning.
session is very fruitful session, ma'am. I
Thank you.
asked one question. Future scope of biotech in artificial intelligence role, ma'am.
That is also a very important thing now everywhere. Like even now the newly research sanctioned our research park in MS University is a bio IT research park. Now everywhere you can't separate this is IT, this is biology. So artificial intelligence in pharmaceutical and biopharmaceutical is also a very important domain. We are getting more opportunities. Say for example, like data science, everything is in data. So if you have lakhs of molecules, everything is now documented and converted as a data science, as a digital science, so that our time and wasting of time in number of unnecessary molecules will be reduced. So as you rightly said, artificial intelligence in pharma as well as the biotech division is have more opportunities. And now people are also interested in developing some bots, like robotics. They support the scientists. They support the scientists in making you some reagents for these are all some of the people. They are hot topic of some Western countries. They are developing the robots. Even in China, some of our companies, I can see that they have robots to make the reagents. They can prepare reagents, thousands of reagents within 1 hour. That humans cannot do. There will be a manual error. So the error is get eliminated there. Time consumption is very less. So definitely AI, artificial intelligence in biotech as well as the pharma have a big opportunity.
Yeah. Vineet.
Yeah, thank you, Gopi sir. Thank you, Dr. Menaga. Very nice session. Thank you very much. In one of your slides, you mentioned this difference between bioreactors and fermenters. So, I would like to ask: are there any bioreactor manufacturers in India?
Definitely, there are some manufacturers out there. I can even give you some details later. There are some manufacturers, bioreactors manufacturers, local manufacturers is also there. So, but what is your product you are looking out? Based on that, you have to go with the reactor.
Okay, yeah, thank you.
Chetan. Chetan had a question. Chetan.
I am very happy. Like it cannot be a question and answer. I am also getting more knowledge from you. I am also a very small startup company owner. So I am looking forward to get more information like Dr. Kannabran sir and another person. The Dr. Kannabran sir, the information is very. I was like now thinking developing a strain in a high temperature. Definitely, I will enter into that area also with our expert team.
Ma'am, I'm Chetan. Are you getting my voice?
Yes, sir. Yes.
Yeah, please.
Ma'am, I want to ask if some of the I work for R&D, product development. So for some of the thing, I do not have a facility for the fermentation. So can we plan and trial at your end? Do you offer that?
Can we plan? I'm sorry, voice is
Trials or the development at
trial.
your
Yes,
end?
yes, sir. Definitely. I have given my mail ID. You can write in the Dr. M Menaga 81@gmail.com is my personal mail ID. bd@bionimtech.com is there. Definitely, you can write a mail. We will have, we can have a one-to-one discussion. And I am always open to experts to come out with good, tie-up.
Okay.
We have a lab fermenter. We have a lab fermenter for making proof of concept, and also we have 1,000-liter fermenter, 2,000-liter fermenter, so that up to commercial production also we can support the people. For R&D, we have a lab fermenter, and experts are there to give some solution to your challenges you are facing in the industry.
Okay, ma'am. Yeah, thank you.
Thank you.
Raivo, please go on.
Sir, actually, this is one thought which came into my mind. The sir said that the major cost for production in this kind of industry, electrical or the electrical cost. So if ma'am's industry is into some biotech park, why don't they put their own captive power plant? Say for 250 crore is required for one unit, and for 1 megawatt power, you require 5 crore rupees to put up a power plant. So is it possible, means anybody thought on this? If we use the captive power plant, our power cost will reduce. Generally, we get power as a 6 or 7 rupees per unit. And if you produce the power there itself, then it will reduce down to 3 rupees per unit, all inclusive.
Sir, this is where we are residing as a women's biotech park. We are, the biotech park is started in 2002 or something else, I think so. Yeah. But now they don't have a facility of, giving place even to install a power plant. So that, that's a small, there are some restrictions for making use of some of the schemes, government schemes in our park itself. But definitely, as I said, they have a central government project is sanctioned for making a, park for fermentation-based products. I am looking forward to the government, to the support of the government, so that they see, making a power plant by ourselves, making everything by ourselves, then what is the use of government to support us, right? So there should be some domain where government to support us, and we can definitely concentrate on our manufacturing process as well as our exports. So I think it should be, Women's Biotech Park is not a place where there are some restrictions are there. Since it's a old park with less space availability, people are there to support us, but less space availability. We are looking forward to go outside and, go for a good, like this type of special park. I want to associate with this.
Okay, thank you, ma'am.
Thank you.
Sanjay?
Yes, sir. I would like to, Madam, it was a good exposure to, technology which we are not familiar with. But I was looking at the market share, Madam. You mentioned that the alcohol market share is over 50%, right? 52 point something. And the second chart you showed that some 22 items are there which need to be imported for medicinal. I think most of them are medicinal, right?
Yes, sir.
Yes. Now, and you also mentioned about the domestic sourcing, 20 to 40%, where which I understand is basically the potential, right? Because 100%, like first one you take, 100% is imported, and against that domestic sourcing is 20 to 40% is the potential which we have, right?
Yes, sir. Yes.
So
domestic source. Yeah.
So we can reduce this 100% to 60% over years.
Over years. Yes.
Okay, so
that's why government is supporting. If we are entering into this particular 22, now what are all the list they have given? If we are starting a manufacturing unit on making this product, they will give the capital subsidy also.
Okay. Now, overall, if you see between alcohol and medicine, both one is need. Medicine is a need, other is a habit. But the CAGR is growing in beverages side, like alcohol, by 6% or 6.5% or something. So what do you think is the market share going to be more? It will be medicines or it will be beverages, which is more intuitive, it will be more beverages. So, what do you say about that?
Yeah, sir. Actually, even though you are rightly said, it's a habitual. The alcohol industry is a habitual. Not only we should concentrate mainly, it is also used in some food industry also. Some of the thing they are using in the baking and all, the yeast is used in the ethanol production is also used there. So it should be like we have to take the alcohol industry as well as the food industry. But coming to the medicine, you are rightly said, you can see that the COVID period, like in the last 2-3 years of pandemic period, there is a big shock, big downflow of this, beverage industry. There is only a peak in the medicine industry. So I think my suggestion is, my thing is, medicinal is a more, opportunistic one. We have to concentrate more on that.
Yeah, we need to support more medicine.
Support more on the medicine. Since here you can see that the 21st, the streptomycin, that streptomycin, you all already know that without streptomycin formulation product, there is no, any type of antibacterial, antifungal, or your product tablet or syrup you are taking, you can see this type of streptomycin thing will be there. But still that we are importing. You see the 100% of dependency is import there. So where we are now, we are always importing and making a good product. So what is the use of making only the final formulation in India? Government should support us in making our own manufacturing sector here itself. As already sir said, the penicillin, he even entered into the penicillin production and got failed. So where we are now? So as a young generation, we are looking forward the government to support us, not only in the production. There's a strong R&D team also.
So what you are saying is the good taxes which they collect from alcohol should be used for subsidizing medicines, right?
Definitely, there can be.
Okay. And
thanks for the idea.
And the second question is it is said that wherever you can make medicines like antibiotics, because it inhibits the other organisms, right? So wherever medicine is a possibility, toxins are also a possibility. Toxins, yes, which could be with high potency, or which could even spread also. A small amount can actually harm a lot of population. I mean, when you mentioned about COVID, it comes out of that only. I mean, not exactly in that spread, but in a different way. So can it be used in for wrong purposes, in wrong hands? I mean, I'm not talking about biowarfare, but is there any regulation, or is there any kind of oversight for that, or do you think that it is we are at a very initial stage now, that we don't have to think about all these things now? Is it?
No, I didn't get your actual question, sir.
Question is, like, wherever medicines can be made, like if you have a technology to make something which heals the body, you can also make a technology which can harm the body.
Okay.
And harming is not localized, but it could be in a large scale as well. I mean, anyway, we are at two initial stages possibly, but there is always a possibility. Like COVID.
Yeah, that's what there is. Regulations are there now, sir. Even if you are saying that I have a plant, I have a GMP plant to produce a particular product, and that is also inspected a team of people, whether you are doing that product safely, whether everything that is called the DM of what we are doing. So everything is get documented and inspection is there. They are approving the plant, and in that plant you can do only that particular product. Okay. Say for example, you can't get an approval for making product A, and then you can't do the product B. So it's unethical, and if you have started doing that type of it will be easily caught by the standardization agencies.
Understood. Okay, yeah. Yes, ma'am. Thank you very much, ma'am.
Thank you, sir. Yeah.
Dr. Kadiravan.
I think the very good feedback from the people. I think Annamalai Alumni Business Association has to definitely take this feedback to the government and support us. I am asking Mr. S.D. Gopi sir who can take this feedback to the ministry and get us some subsidies in power, support for R&D and all.
Parliament, will do, man. Okay. Dr. Kadiravan. Okay, I think there's anyone else has a question? Please unmute and ask. Sir, Dhanasekaran, sir.
So there are some questions in the chats also. Thanks to Varadarajan, very good session and active participation. Thank you. And do you provide facility for fermentation product styles? Yes, we have a facility for making products such as kombucha. So Chetan, sir, yeah, you can definitely come out and we can have a discussion. So thanks.
In fact, Mr. Varadarajan is from Lansatech.
Okay.
He's a country head.
Different zone.
Okay.
Is there, Dr. Kadiravan, sir, is there any fermenter for plant root growth production? Definitely, I will look it over, sir. And he can also connect with me, Dr. Kadiravan, sir. He can think about this plant root growth also.
Sure. Anyone else has a question?
Yeah, sir.
Okay, we are done. We're done with the questions now.
Yes.
Thank you.
Okay.
Thank you very much, Dr. Menaga.
Thank you, sir. Thank you
Thanks,
so much.
Thank you very much for taking your time out on Sunday morning to be with us and address us.
This is a very informative session. Actually, I have to thank all the participants because you have thrown the torch for thinking of some other 2-3 lines for making an R&D much more better in the fermentation. And I'm looking forward to participate in the next sessions also. Definitely.
Oh, sure. Thank you. Thank you.
Thank you.
Take
Thank
care.
you all. Thank you so much. Yeah.